Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. If you're finding our deep dives valuable, please take a moment to like this video, subscribe to our channel, and share it with your friends and relatives. It really helps support the Sand Science Experience and allows us to bring you more incredible science. Today, we're diving into something pretty incredible, really groundbreaking stuff in xenotransplantation. Humanity's always pushing boundaries, and the organ shortage, well, it's a massive one, affects millions. So we're going to unpack the very first case of a genetically engineered pig lung being transplanted into a human. Okay, let's get into this. The organ shortage crisis, it's huge, right? A persistent challenge. And scientists, they've been looking at xenotransplantation using animal organs for ages. But lungs seem to have been especially tricky compared to, say, hearts or kidneys recently. Why is this lung study such a big deal right now?
Yeah, that's a really great question. You've hit the nail on the head. While we've seen some, pretty impressive progress with hearts and kidneys lately, the lung has always been lagging behind. It's just inherently more complex. Think about it. You've got incredibly high blood flow going through it constantly. Plus, it's directly exposed to the outside air every single breath.
Yeah.
Bacteria, viruses, allergens.
Right, a direct interface.
Exactly. And its structure is so delicate, it's just super vulnerable to an immediate and really aggressive immune attack from the recipient's body. So this study isn't just, another small step. It's genuinely the first time a pig lung has shown it can remain viable in a human body. That's overcoming a massive historical hurdle.
Wow, okay. That puts it in perspective. A huge barrier cleared. So this is where it gets really fascinating for me. How? How did they actually engineer a pig lung so it could survive? What kind of pig was it? And what was the biological tweak that made it possible?
Right. The how is all about some very clever genetic engineering. They used a lung from a specific type of pig, a Chinese mama shang pig. And this pig wasn't just any pig. It had 6 specific gene edits.
6 edits?
Yeah. Think of it like this. The human immune system has like a most wanted list for foreign tissues, especially pig tissues. So first, they knocked out 3 pig genes. These are genes that produce certain sugars on the surface of the pig cells.
The red flags.
Exactly. The things that scream, I'm foreign, to our immune system and trigger that immediate rejection. They got rid of those. But then they also added 3 human protective genes. These act more like peacekeepers, maybe.
Okay.
They help regulate parts of the human immune system, specifically the complement system,
Really?
and also help prevent blood clotting within the transplanted lung. Both are absolutely critical for survival.
So removing the triggers and adding some human camouflage and protection.
That's a good way to put it, yeah. And this specially engineered lung was then transplanted into a 39-year-old male recipient who was declared brain dead.
Incredible precision. Just astonishing. So you have this incredibly prepared organ, this complex procedure. What happened next? What were the results? How long did this lung actually function? What was the biggest win, but also what were the remaining challenges?
Well, the biggest success, hands down, was the duration and the lack of hyperacute rejection. The lung remained viable and functional for 216 hours. That's 9 full days.
9 days. Wow.
Yeah. And crucially, without signs of that hyperacute rejection, the immediate catastrophic attack we used to see within minutes or hours, avoiding that was huge.
Okay, so that part of the engineering worked.
It seems so. But it wasn't all smooth sailing. Challenges definitely emerged. Within about 24 hours, they saw severe edema that's swelling in the lung. It looked a lot like something called primary graft dysfunction, which sometimes happens even in human transplants.
Right, when the organ's been stressed.
Exactly. And then later, around day 3 and day 6, they started seeing signs of antibody-mediated rejection. This is the body's sort of slower, more targeted immune response kicking in. Ah, okay. So the initial defenses were down, but the next wave started.
Sort of. They saw antibodies building up, things like IgG and later IgM, and activation of complement C4. Now, the team was using a pretty heavy-duty immunosuppression regimen, a cocktail of drugs, to try to manage all this. And there was some evidence of recovery by day 9. Also important, they monitored very closely for infections, especially pig viruses, and confirmed no active infection despite finding low levels of some porcine viruses.
It really sounds like a constant biological negotiation, doesn't it, between the engineered organ and the human system. So pulling all this together, what's the big picture here? What does this deep dive really mean for medicine's future? What's the main takeaway?
I think the main takeaway is that this is a truly critical step forward. It's more than just one successful, albeit temporary, transplant. It gives us absolutely invaluable direct data on how the human immune system actually reacts to a pig lung xenograft in real time. Stuff we just couldn't know for sure before.
Real-world data, essentially.
Yeah.
Exactly. Now, obviously, challenges remain. Figuring out how to better manage that early graft dysfunction, that edema, and fine-tuning the immunosuppression, maybe needing different drugs, perhaps adding things like CD40L blockers, which are showing promise elsewhere,
Right.
and of course needing longer-term studies. 9 days is amazing, but we need weeks, months, years eventually. But this research, it really does pave the way. It shows proof of concept and gives crucial direction for the next round of genetic refinements and drug strategies.
So it cracks the door open, even if there's still a way to go.
Definitely. It makes the prospect of using xenotransplantation to solve the organ shortage feel, well, significantly closer.
This deep dive really illustrates just how close we might be getting to a future where organ shortages could become a thing of the past. I mean, just imagine the impact that could have on countless lives. Real hope where maybe there wasn't much before. What new questions does this incredible research spark for you about the future of medicine? How might this reshape things? Something to think about. And please don't forget to like this video, subscribe to our channel, and share it to support the science experience. We're really grateful for your support.